start with introducing Brigitte, who is presenting the project Alternative. So Brigitte has obtained her master's in pharmaceutical sciences and PhD in neuropharmacology from Vijay Universiteit Brussels. She has joined Cinciano in 2010 and she is currently senior toxicologist in team leader in risk and health impact assessment service. She coordinates and participates in multiple national and international research projects and activities in the area of geotoxicity and chemical agents, which are typically focused on food contaminants and developing and employing new approach and methodologies. She participates in various national and international working groups, scientific committees that are on hazard and risk assessment. And she is currently the president of Belgian Environmental Metagenesis Society. She is also a lecturer in toxicology courses of different Belgium universities. So with that, Brigitte.
Cells, and on the other hand, we have human-induced pluripotent stem cell-derived cardiomyocytes. And so these three components form together the 3D bioengineered myocardial tissue model. On this slide, you can see some more details on the scaffold, and I will also show you some more information on the hydrogel. However, I will not elaborate too much on this in case you have some questions. Gianluca Ciardelli is also present here today, so if you want to have some more information, I think he will be happy to answer that. Maybe also one thing that is important is that different types of scaffolds were investigated, so both isotropic and anisotropic, and if I'm correctly, it was in the end one of the anisotropic, so one of these two that was used to develop the final 3D system. So then we also need the hydrogel, and this consisted of gelma. You can see the synthesis over here. So there was then gelma solubilization in aqueous media, and then the chemical cross-linking was done through light irradiation. What is also important is that tuning was done with several percentages of hydrogel, because the percentage of hydrogel will define in the end the stiffness of the tissue. And this is important because we wanted to mimic both the young and the old cardiac tissue. And in order to do that, two types of systems were developed. So one containing 5% of the gelma, and this was reflecting the young cardiac tissue, whereas there was another model containing 10% of the gelma, which has this an increased stiffness, and this mimics then the old or the aged cardiac tissue. And then, as indicated, of course, we also need the cells to be part of the final system. And so two different cell types were used, as I mentioned before. So on one hand, the cardiomyocytes, and here you can see the procedure that was used for the maturation of the cardiomyocytes. And you can see then that at the end, the cardiomyocytes are showing some contractility, so to mimic, in fact, the beating of the heart. And then these cardiomyocytes were then combined with the human coronary artery endothelial cells. So all components were then put together in a model, and then this model was molecularly characterized. So different markers were evaluated, markers with respect to cell maturation, contractility, and so on. And overall, you can see here on the slides, without going into the details, that the 3D system, so both the young and the old, showed clear differences in the different markers compared to the 2D system. The 2D system, which was used as a point of reference, because previously the research was done with the 2D systems, but you can see that by using these 3D models, this has an important impact on the markers, and that you can also see that there are clear differences between the young and the old cardiac tissue. So once then this system was put in place, we of course also wanted to start testing chemicals in the system. And I want to highlight here that chemical selection required quite some discussion, and we took our time to really come to an appropriate selection of chemicals, because different aspects needed to be considered. So first of all, we needed to have evidence, of course, that the compound was inducing cardiotoxicity. So in order to have more insights on that, we used the literature review that was performed within the context of alternative. So in fact, 2 literature reviews were performed, one collecting the evidence on a potential association between exposure to toxic environmental chemicals and left ventricular dysfunction, so one specific type of cardiovascular disorder. And we found, in fact, based on this study, that most evidence is currently available for such an association for particulate matter 2.5 and 10 and arsenic and lead. However, as I will discuss also later in this presentation, we need, of course, also to work with chemicals that are relatively easy to put into our system, and the particulate matter and the metals are not that easy to work with. So that's why in the end we decided to focus on other chemicals to perform our testing on. And therefore we also took into account toxicological evidence, so data from in vitro and in vivo studies that were available on compounds that had already been assessed for their cardiotoxic potential. Then secondly, we also took into account the mode of action of the chemicals, because of course it's important that the mode of actions through which the chemicals are leading to cardiotoxicity, that they can be picked up in the system. And in order to help us with that, we took into account the adverse outcome pathway framework. So within the alternative project, we also spent quite some time on developing an AOP framework for cardiotoxicity. And for those of you who are maybe not familiar with the AOP concept, so the idea is to have a kind of framework, so the AOP framework that can help us to structure the knowledge, because within this AOP framework, you're not going to describe in detail everything that is happening between the molecular level and then the observed adverse outcome, but instead you're going to focus on some key events. So what we did is, in fact, we combined already available AOPs related to cardiotoxicity, which are available in the AOP wiki. And then we combined this with newly developed AOPs. And for these newly developed AOPs, we used the evidence that was then also collected in the systematic reviews, collecting toxicological and epidemiological evidence. And then based on that, we chose to focus on compounds leading to mitochondrial dysfunction, because mitochondrial dysfunction, so the interference with the mitochondrial complexes, is also something that can be modeled with QSAR models. And this is, I will come back to that later, also an aspect that was done within the project. And I think this also nicely shows that this kind of AOPs can help us really to give us more insights in the type of information that a system is providing us, how this in the end leads to the adverse outcome. So then I already just referred to it. So we also wanted to take into account that we would do some in silico modeling on these compounds. So I mentioned we had taken mitochondrial dysfunction, because here we can also develop some QSAR models for the molecular initiating events. And on the other hand, we also wanted to do modeling to do the extrapolation from the in vitro setting to the in vivo setting. But therefore, we also needed some kinetic data on the compounds that we selected. And then finally, we also had to make sure that the compounds that we selected were compatible with the in vitro test and the omics analysis that we would perform. And this is, of course, important with respect to, for example, physicochemical properties. You need to be able to bring your compound into solution, and also some laboratory requirements. For example, preferably you also work with compounds that are not too toxic, although, of course, if you're performing toxicity testing, this is something that you have to deal with as well. So taking all this into account, we in the end ended up with 3 chemicals of interest, and they are mentioned here on this slide. And you will see that 2 of them, so amitriptyline and doxorubicin, are 2 pharmaceuticals. And this, one of the reasons is, of course, that for pharmaceuticals we have much more data available. So in that sense, it's easier, certainly in the beginning when you're developing a test system, to work with data-rich compounds. So you can see here the first amitriptyline. You can find some information on the pharmacological effects, also on how it's leading to cardiotoxicity. And then doxorubicin, which is in fact now considered a little bit as the golden standard for cardiotoxicity. And here again, you can see its mechanism of action as well as its cardiotoxicity, how it is executing this effect. And then as a third compound, we included rotenone. Rotenone is naturally present in certain plants, but it's also commonly used as an insecticide and pesticide. And it's also known to lead to cardiotoxicity by interference with the mitochondria. So that's why in the end, we ended up with a selection of these three compounds. And importantly, we also included a negative control for cardiotoxicity, which was sodium saccharin. So then we did some testing with these compounds. Not everything has yet been finalized, so I will focus now on this part on the results, some of the results that we obtained with doxorubicin. Here you can see the outcome that we had with the test with the doxorubicin in the 2D system and the 3D system, but only the 3D system with the young cardiac tissue. And without going too much into the details, you can see that both with respect to cell viability and the generation of reactive oxygen species, that there is a clear difference between the 2D system and the 3D system. Then another important part was the performance of omics experiments. So I will show you also now the data with doxorubicin. So doxorubicin exposure was done for 1 week and then with different concentrations, and then the data, the material was collected to do 3 types of analysis, including metabolomics as well as proteomics and transcriptomics. And here on the right, you can see the results that were obtained in the, first of all, in the 2D system, because we didn't have yet data for doxorubicin in 2D system. So we first wanted to have some more insights in this. And you can see that the metabolomics results show that there is a distinction between the different concentrations of doxorubicin, and you see that at higher concentrations we are further away from the control, indicating that indeed several things at the level of the amino acids are going on. Then we had also the results of some transcriptomics work that was done with the doxorubicin. You can see it here on the left. Without again going too much into the details, you can see that several pathways were clearly affected after exposure to doxorubicin. And maybe important to mention also that with saccharin, this was not the case, so confirming indeed that what we are seeing was not happening with every compound, but specifically with our compound of interest. And then you can also see here the results of some metabolomics outcome, where we saw, for example, that the metabolic pathways were clearly affected. So then this work was then also repeated in the 3D system. There we compared how the 3D system was responding to one specific dose of doxorubicin. And then in that sense, we wanted to compare the young tissue versus the old tissue. The exposure was also for 1 week. So we looked at after 1 week what was going on. And you can see here on this plot, so summarizing the outcome of the metabolomics, because the results for the transcriptomics and the proteomics are still being processed. You can see that there is a clear distinction between the markers in the young and the old tissue. Now, if we go a little bit more into detail, we see, for example, that anserin, serotonin, and kynurenine are increased in the old versus the young tissue. As these markers are typically for the cardiac tissue where there are problems, where things are going wrong. So it might maybe that our aged tissue is reflecting more also situation where there are some problems where there is some damage. So this is certainly something that will be explored further. And the results will also be combined with the results of the other chemicals, so that will allow us to hopefully identify some markers specifically then for cardiotoxicity. So then to conclude, so again, I would like to highlight that by developing an AOP network based on systematic review of toxicological and epidemiological evidence, this can also give us a tool to really better understand, to better situate where the type of information generated by a more complex system as we are dealing with in the alternative project, which type of information it is providing us, and as such can also help regulators to know how to deal with this type of knowledge. So in the end, so we have our in vitro 3D system, but it will be combined, as I mentioned, with in silico models in the end to come to a kind of platform that helps us to mimic heart physiology. By integrating also the omics, this can help us to give us a global picture and hopefully also to have some markers on which we can then focus, so some specific markers for cardiotoxicity. And finally, what's of course also very important is that since we are dealing with an in vitro system, we need to have a way to connect the in vitro data to the in vivo situation. And there, I didn't show this in detail in this presentation, but there also work is being done on developing this type of PBK models to really help also to translate the in vitro information to the in vivo setting. And so with this, I would like to end the presentation. Of course, thank all the partners involved. And if there are some questions, I will do my best to answer them, although I might not be able to give you all the details, but we can certainly address this and also later if you're interested.
Yeah, I think now thank you, Brigitte, for this insights. And now the floor is open for question. Any questions, anyone? We also have our project coordinator, Gianluca, here in the meeting. So we all can together support if there are any questions. Okay, I think then let's open the floor for more questions at the end when we dive into the workshop and take it more deeply. So now I would like to go the second presentation that will be from the project LIFESAVER, and we have Michael Gassett to present it. Just introduction from him. So he is chairman and co-founder of Sequira Limited in Helsinki, Finland, and he's also the ambassador in Finland for European Orthopedic Research Society. He is full-time professor at Aalto University Foundation, Espoo, Finland, and has visiting professorship in UK, Japan, Italy, Portugal, and Estonia. With over 300 publications and patents spanning across 30 years, he has led numerous EU, IEA, and national projects. Michael was also appointed at EcoRS Ambassador for Finland in 2020 and has several presentations in international committees and organizations that also includes Cost Actions and European Connected Health Alliance. He specializes in developing innovative methodologies, in vitro testing methods, and owns LinkedIn group for advanced testing of biomaterials. So if anyone is interested, can also join that LinkedIn group. And Michael founded Sequira in 2016, which is focusing on biomaterials, medical devices, and advanced therapy medicinal products. And they are into development in compliance with regulations like MDR 2017 and IV MDR 2017 746. So thank you, Michael, for being here and joining us and collaborating with us. So the stage is yours. please enlighten us on your work in the project LIFESAVER.
Thank you, Pooja. It's always nice to hear what I have been done, or you have been done. There's this thing, did I really did all of that stuff? Okay, thank you. So I tried to share my screen. I don't know if you have seen that.
We can see your screen, Michael, but unfortunately the content is not visible. So, Brigitte, would you mind sharing?
Yes, I can share. Yeah.
Okay, I
maybe
stopped
Michael,
sharing
you stop sharing. Yeah.
It's yeah. It also show I stopped sharing now. Okay. And maybe if you can open my presentation. So I need to say then next slide.
Yes, I'm trying, but I also have an error message here. I think I was too fast.
I'm not sure why sometimes the technology decides to surprise ourselves when we are really wanting it,
Okay.
I think it should be okay now.
Yeah, now
Just
now
let
I
me
can
know
see. Yeah,
if I
perfect,
have to
Thank
go
you.
to the next one.
Thank you, Birgit. So you will be helping me to move on. So thank you everyone for coming to this webinar. And I presenting here on behalf of the consortium the brief outlook on the LIFESAVER project, which is concerning the assessment of the risk of drugs and chemicals for fetal health, or actually for fetal maternal health. Next slide, please. So we will focus here today more about the problem and the impact rather than to talk about the technology. And the driving force for our project, similar to Alternative in this call, has been that basically we are missing data on the safety of medicals and especially environmental pollutants as well for pregnant women. There are no clinical trials going on, very little, and these which are going, they are very, very, how to say, limited in terms of that something is still going on, something is not clear, so that we don't have really the information, which is very important part of that one. So next slide, please. And we have here collecting first when we started proposal to write a lot of information about what has been done in the world. Most of these publications come from US. And eventually there is a trend also that in some cases they said that it's not ethical not to include pregnant women in clinical trials because they remains a vulnerable population or sensitive population which does not have a proper drugs. So in respect to the dose, in respect to the selection of the compound, and in respect to the therapeutic protocol, how this thing should be done. So the starting point for us was a lack of information. And the next slide is, please, thank you. We also know that the clinical trials is very difficult. The problem, especially in the pregnancy, is that there is no animal model which can sufficiently replicate human placenta, which is significantly different from that what we have in primates, or significantly different what we have in cormal models. So it's very difficult to replicate the placental functionality as such. And in this case, the translation from that placenta to humans even more difficult. To give you just an example, the blood flow in animals in the placenta is different from the blood flow in the human placenta. And going to the project further is that what we have seen also that even before talking toxicology, before talking the effect of certain compound on fetal health or maternal health, we need to know the exposure. We need to know how much of specific compound is passing the placental tissues and how much it going through that barrier, which separates fetal and maternal blood flow in certain amount of time at certain amount of dose. Next slide, please. So the goals of the project was to find and create a reliable and reproducible relevant methodology to analyze and predict the transport of the substances. So we are not dealing with toxicology. We are not trying to find out the pathway or something, because that's a completely different story. What we try to find an answer for is to say how much drug will penetrate the placenta if the dose is taking like that, and the time which pass after that one is taking like that. And to do that, we are developing the biodigital twin system, which may sound a little bit a new concept. And based on that biodigital twin, we plan to get a regulatory advice and policy advice how these issues needs to be solved. Next slide, please. So the concept of the project stays in the combination of the biodigital twin, which is in vitro in silico parts. And we have now 2 in silico models, which one is bridging the in vitro part to the real 3D placenta model, which will be able to use a special software tools and experimental data to make a prediction, leading to possible policy advice and regulatory advances in this field. So, of course, that if we would be able to do that, or maybe better to say when we will be able to do that, then we will get a huge impact on the society, not only in Europe, but basically worldwide, because we have 200, in Europe, like 5.5 pregnancies per year, and in the whole world is like the 3-4 times bigger amount of that one. So next slide, please. So the approach what we have is that we start in our work packages, we start with the pregnancy biology, the boundary conditions. It was a little bit surprising that we find that how many data are missing. There are basically no knowledge in the end of the first trimester pregnancy, what the placenta is actually having, what is the blood flow, what is the pressure of the blood, how many seconds the blood actually stays in the placental and maternal volume, how fast the diffusion happens through the Willows wall, what is a Willows wall composition, how thick is it, and so on. So based on that, we're able to collect some database, and then we do our best available knowledge to manufacture the in vitro testing system, which is a microfluidic platform based with a specific chip which simulate the Willows structure of the membrane, which is a bioprinted membrane. And based on that, the 2 in silico models, which is describing what happens in the chip, and the second model, how do you translate this data to the 3D placenta, because then you can be able to simulate where exactly your drug is located in space and time. And based on that, we will be able to predict and to provide to the regulators the data about that, how safe that specific drug or compound is in pregnancy compared to some controls. So for example, is it fast penetrating or is it slowly penetrating the tissues? Because even if there is fast penetrating, but it is not toxic, maybe that's not a big problem. On the other hand, if it's slowly or very badly penetrating the placenta, which may be considered as safe from point of view of the application, but if it is highly toxic, then it's another story. And of course, we also have the exposure, we have dose, we have time. So time, dose response is extremely important to provide the proper correlations. Then we are planning, we have a special work package on EU policy advice, which we now work with the regulators to produce a certain protocol which could be used for estimation of any drug or pharmaceutical product or chemical whenever imported, produced, or exported in the European Union, which will get a great implication to their clinical relevance, because that, well, if we have a high ambition that, let's say that any instruction for use in every package in every blister has to have the data saying that is it a highly penetratable or is it slowly penetratable. Next slide, please. So the analysis example, what we do is just to, as I said, we are not looking for toxicology here, but what we're looking in engineering sense is that we have some input on administration side or maternal. We are not considered specific mechanisms. There are a lot of them, but what we can see the concentration of the drug or metabolic product of this drug on another side of the membrane. And that gives us a possibility to reduce significantly animal test and clinical test. And basically for every clinical test which involve pregnant women, that is an extra 5 million euros, which need to be invested to have the extra regulations and extra permissions and all extra. We can process retrospective or digitized data. So if somebody has it done in another way. And basically we can do it for virtual LNA assays and can be linked to therapeutic approach. And that what has been submitted in a patent application, the combination of these transport properties would result in a parameter or set of parameters we call drug transport signature, which is a unique combination for the specific drug in relation to penetration to the placental tissues. So that does not need us to know the kinetic or pharmacokinetic model. Of course, it's nice to have it, but then here, as I mentioned again, we not talk about the pharmacokinetics, we talk about how the drug is transported through the placental tissues. And altogether, we plan to provide a thing, what we call IREAD, which is independent risk of exposure assessment data, which needs to be quantified and submitted together with their drug development purpose. So next slide, please. So that the second part of the project is also concerning the risk assessment and corrective actions, and also the quality procedure which is required by regulation in pharmaceutical applications, to think that how well the pharmaceutical production would address whether this or that specific drug can be considered to be safe in pregnancy from the point of view of penetration or not. So that gives an additional security to the doctor to prescribe or not to prescribe certain drugs, because also despite of positive negative discrimination, so something should not be used, we also have positive discrimination, because maybe there are some drugs which needs to penetrate the placenta because there can be different comorbidities and different diseases. Next slide, please. So our vision is that we would like, maybe not this year, but next year and after next year, to create a worldwide standard for preclinical risk assessment on potential exposure, to providing IREAD numbers for the specific drugs and combinations, and say that that basically is what you need to know whether this specific compound is penetrating placenta or not, which eventually affects 200 million pregnant women every year for the whole world. Next slide, please. And this is thank you to all our consortium, which you have seen now the partners and the map, how we are distributed all around the Europe. And we are entering now the last year of the project, which will be finished at the end of 2025. We basically plan already to demonstrate the system. We demonstrate some component of that, but the main demonstration will be done next year. And I think the next slide will be the last one. So thank you. You can freely follow up. You have the link already in the chat. You can follow us on any social media you have. We have very nice videos. We have blogs. We have posts. We have newsletters and everything you need to know if you're interested more. And for any interested partner who would like to cooperate, we can announce a pilot project, which we will start after the formal project will be concluded in the end of 2025. Thank you. Thank you, Birgit, to handling that.
You're welcome.
Thank you, Michelle, for enlightening us about the work being done in the project LifeSaver. So I can see that there is a hand from Martin.
Yes.
Yes, thank you very much for this very clear presentation, really exciting work, and really interesting to see that, again, the development of non-animal methods is triggered in a field where obviously the animal model is not so great. Anyway, I would be interested to know, there's probably a time course in the development of the placenta during pregnancy, so maybe quite different from the beginning to the end. And do you focus on a certain period? I've understood
Yes.
it's rather early phase.
Yes, 12 to 14 weeks.
Okay, and is there a specific reason that you focus on that
Yes.
early phase? Okay.
Yeah, because this is a reason when the end of the first trimester, where we have transition from embryo to fetus, where all the mature components, on the fetus start to be built. So we talk about neural system, vascular system, all these kind of things. And I think that is, we find out in the literature also the mature complications which come later in pregnancy, they affected at the end of first trimester, beginning of the second trimester. And this is one of the things why we can't really replicate it from for example, from the placenta after delivery, because the placenta at the delivery is totally different.
Okay, thank you. So you also don't foresee to extend this placenta model to other stages of pregnancy?
It's technically possible, but
Mhm.
it requires a little bit more effort. In this project, we only focus on that time, because we have limited time and resources as well.
Of course, yeah. Thank
So,
you.
Birgit has a question now.
Yes, I also
yes.
had a brief question. Thank you very much for your clear talk. If I understood correctly, the focus is really on pharmaceuticals, but I can imagine that if you have such a system, it can also help to give us more insight to what extent, for example, chemicals in general are passing the placenta.
Yes,
Is this something you consider as well, or?
Yes, it's in the plan. So we have a list of compounds. We start to do that. They include antibiotics, antiviral preparations, hormones, dimethyl phthalate, and bisphenol.
Okay, yeah. So you will include
Everything all you can mix it together. We find out, of course, because we have also limited amount of time for experiments, that maybe some of that combinations, because the commission is especially interested in the mixtures, and some of them, they may not have therapeutic relevance, because doctor usually does not prescribe antibiotics and antiviral in the same package. So, however, what I think that if we have like doxycycline and estradiol, like environmental pollutant, because we have a lot of hormonal pollutions in environment, that may be one of the example of the mixture which we can test.
Thank you.
Great. So looks like already that the floor is open and we are ready to discuss more. Let's formally introduce Martin also, since he will be joining the panel. So Martin is a trained toxicologist with more than 20 plus years of experience in regulatory toxicology and non-animal methods. He's leading the regulatory science group at MUI, working on development of new toxicological assessment approaches for regulation within his role as Austrian national delegate for several OECD experts. And the groups include WNT, DNT-ITA, VMG-ECO, ESCA, and so on. And he is also involved heavily in PAPRE network. Furthermore, he works for European Food Safety Authorities as a member of Plant Production Products and Residues (PPR) panel and other EFSA working groups that are related to adverse outcome pathways based on integrated approaches to testing and assessment. He's also working for transformation to an animal-free regulatory system through building bridges between science and regulation. On his work presents and group presents a regulatory science partner within the European research projects like Alternative, Chiasma, Insight, and PRC. He's also leading EFSA-funded DNT RAP4 project, providing 4 new case studies for the use of DNT in vitro test battery for regulation of pesticides. He's also leading a proposal on EPAA designation challenge for development of new fully and non-animal based methods based hazard classification system. And for the project alternative and the European Union, he's also developed a sustainable policy on using sustainable methods in terms of conservation, in terms of energy, etc. And here the floor is yours, Martin. Welcome to this panel, and we are glad to have you here. Next, we are also joined in the panel by Laura Lecolsi. So she is a senior consultant in innovation and competitiveness at Idea Consult. Laura has brought interest in entrepreneurship, innovation, impact measurement, and evaluation. She has worked on different impact assessment and evaluation studies, both at regional, that is Flemish, and European level. Currently, Laura is involved in evaluation study on excellent science in European framework programs, which include research and innovation on behalf of DG RTD. At European level, Laura has also contributed to other studies, knowledge systems in new era, study on monitoring the performance of EU industries and industrial ecosystems, and the LIFESAVER project. So Lara has also been a doctoral researcher and also a postdoctoral researcher at the Ghent University in the field of entrepreneurship and innovation. Laura's doctoral dissertation was awarded the International Heiser Award for Outstanding Doctoral Research in field of new enterprise development. She has published her work in international top-tier journals such as Small Business Economics, Technovation, and Science and Public Policy. So we are glad to have you, Laura, here with us. So now we open the panel discussion on regulatory and technological innovation in health safety. To begin the discussion, we say that we have quite esteemed participants here. So I ask everyone to be open, and if you have any questions, feel free to drop in the chat. We will go one-on-one in a Robin ring fashion. The question will start with Martin, followed by Laura, Brigitte, and Michelle for their comments and opinions. The first question is, so the regulatory and technological innovation, how do you see the project's alternative for Martin and Brigitte and LIFESAVER for Laura and Michelle integrate technological innovations to contribute to regulatory advancement and enhance healthcare safety? So I would like Martin to begin this conversation. Martin, could you unmute yourself, or maybe Brigitte, you want to start with it?
I think Martin has some issues because indeed he was visible before, but now his camera dropped. Yeah. Okay. So maybe I can briefly start, and then I know that Martin has also a lot of ideas on this. But what I would like to emphasize, and what maybe we did not really elaborate upon in detail during the presentation, is that we aim not to just put the one system as such to the regulators, but to put it more in like an integrated strategy, so what we also call an integrated approach to testing and assessment, and shortly an IATA, where you have a kind of combination of different essays that help them to address a specific regulatory question. It also fits into the whole bunch of initiatives that are currently ongoing to move towards this more human relevance and kind of approaches that do not longer directly rely on animal testing, initiatives that we see in like the European Commission roadmap on NAMs. We also have within PARC a lot of such IATAs that are being developed. So this is one of the possible applications that we see. Also, of course, what we are dealing with specifically in alternative, and I heard it also in LIFESAVER, it are kind of more complex systems. And currently in a regulatory setting, from pharmaceuticals, it's a bit different, but certainly for chemicals, our experience with these more complex systems is limited. So we also need to have discussions on how do we need to do the harmonization, how we do the standardization, things like that, and also validation, how should it be done for such kind of more complex systems, because this will of course also be important in the end to have the regulators on board so that they have sufficient confidence in the results that these new, more complex systems are generating. But you can see for the pharmaceuticals, again, they are already a bit more advanced, but for the chemicals, there are also now more and more initiatives that try to contribute to that. But I see that Martin is also online, and I'm sure he wants to add something because I know he has a good overview of all these things.
Just Martin, you got dropped. So the question of discussion here is, how do you think our project Alternative is integrating technological innovations to contribute to regulatory advancements and enhance healthcare safety?
Yeah, thanks a lot. And my apologies, I don't know, my computer shut off suddenly. I don't have a clue why while you were introducing me. So I'm sure Birgit has very well covered this question. I think it became clear from her presentation and probably from her intervention already. So the main tools that Alternative will provide are these more complex cardiac microphysiological methods. So for aged and young heart, we have also new computational approaches for cardiac safety assessment. So to assess especially also mitochondrial dysfunction as an important molecular initiating event. We get new PBK models, so for extrapolating from in vitro to in vivo. We also have new knowledge now available in terms of mechanisms and nicely organized via the AOP framework. And all this will, at the technological level, really help now regulation. I think, as Birgit has mentioned, this needs careful thought on how we go on with validation now of each of these models. So bringing this on the table to the regulators will, of course, again stimulate the question, where do we get the resources from for validation? So we have now additional methods, and this is just a challenge. We need to have sponsors in the field and showing where we would have something, but we need now the sponsoring of further validation, which is not an academic task as such. I think could create impact. And let me say, maybe additionally, so we will go also a bit beyond this cardiac specific approach. So we will develop an IATA so an integrated approach to testing on assessment based on non-animal methods that could be following up, be also applied to other fields, not just cardiac toxicity. So we talk about fundamental principles here. So how to change a regulatory system where we today rely on essentially a series of animal tests that are legally defined, and we take the NOEL from that animal test, and we do the extrapolation to the humans by pragmatic assessment factors. This is how we consider safety, a safe level today, right? And we need to transform this into a new system where we start from a series of in vitro methods and translate this to a human safe dose with PBK modeling. So we do the same extrapolation steps, but we start from somewhere different and we use the different models. And all this needs, let's say, an in-depth uncertainty characterization. Also what we will provide as a package here, some workflow to integrate the uncertainty knowledge, so the uncertainty that comes from this new approach, and to be able to compare this with the uncertainty that we have in the animal tests, as a benchmark. So because this is what needs to happen to bring the uncertainty of the animal tests to the same playing field like the in vitro methods, and to use it as a benchmark to be overcome. Not to predict the animal tests, we will have a different system, I'm sure. We will rely on molecular cellular markers, biomarkers of effect, really, that indicate a risk that something will happen in the real world. And well, so having all this work now, the data, the methods, and the discussion on how to integrate this, I think will also trigger this field of next generation risk assessment evolution.
Thank you, Brigitte and Martin, for giving the perspectives from the project alternative. Now I would like to open it for Laura. We would like to have your views on how the project LifeSaver is integrating technological innovations and contributing to the regulatory advancement.
Okay, thank you, Pooja. So indeed, we at LifeSaver, we are developing complex in vitro systems in combination with in silico components that can contribute to chemical and pharmaceutical safety testing and risk assessment, as Michael already explained in the presentation. So in that sense, the LifeSaver system will be able to enhance healthcare safety. And as the colleagues already mentioned, such new methods, such complex in silico and in vitro methods, they have indeed the opportunity to serve as complementary methods or even as alternative methods to traditional animal testing. And these new complex methods, they have many advantages over traditional methods. For instance, they can be more efficient, they can be faster, less time consuming, they can provide scientifically better evidence, make sure that less animals are used, they allow more human relevant decision making, and so on. So these new methods, they have the potential to contribute to regulatory advancements and healthcare safety, because thanks to these advantages that they have, they can be taken up by regulators and improve regulatory decision making. If, for instance, which chemical is safe, which one is not. But of course, and that is also something that we explore a little bit more, to really spur the uptake of such new methods, in vitro and silico methods, the regulatory and the legislative frameworks, they of course have to be adapted a little bit still to make their integration easier.
Thank you, Laura. Michael, would you like to add something more to it?
I think first Martin has some question.
Maybe if I spontaneously may interact, so I very much like your project and what you have presented, and I could see a very interesting link also to the current focus of the European Food Safety Authority as well as the OECD, where we have a lot of work ongoing for developmental neurotoxicity. So how to assess developmental neurotoxicity in vitro. We have a series of in vitro methods, and we have already identified that PBK modeling, so translating these in vitro concentrations that cause the effect in the in vitro system to a dose that a human, a pregnant woman needs to take to come to that concentration level. So this modeling is quite challenging. And I could see maybe a role here also to bring in here your models. What is of particular interest for discussion for me, so the brain development is relatively, I mean, there's a huge brain phase where is relatively late in the pregnancy. I mean, it starts all over, but there is a peak of, let's say, rather to the end of the pregnancy in terms of growth also of the brain. And so curious to continue that discussion, just to see. I think it's just in time.
Okay, thank you, Martin. I think that we need another workshop to discuss this interesting strategy. So I will continue what Laura was saying, but from a little bit different point of view. Because one of the reasons why we are here today is that we see a coherence between alternative and LifeSaver in terms of the further expansion of that, what you're doing and what we are doing. Because, for example, when you talk and when you find out that about the cardiotoxicity and you know what about the dose, So would you be, for example, interesting to know if the specific drug you're using in cardiology or not to be used in cardiology, how does it affect if the patient who needs to take this drug gets pregnant? Is it something which need to be changed, or is it something that need to be adjusted in terms of dose, or in terms of the drug, or in terms of the compound? And to go to this toxicology and dose response things, you need to know what actually exposure this particular tissue or particular patient or whatever has. So that is basically what LifeSaver was intended to answer first, because we understand, of course, the limitations. You can't, unfortunately, solve everything within one project. So this is one point. The second about regulative aspect is that, as you know, HTIR are coming and next year, and they will concern all medicinal products. So there will be a joint scientific consultation, there will be joint clinical assessment, everything like that, which is supposed to trigger the one window shopping for the pharmaceutical products as well, and as well as medical devices class 3. So I'm talking about 2021/2282 regulation, which is known as health technology assessment protocols. Each is different from that what we have on OECD regarding European Food Safety Authority, because they also have the same problem, as you know, maybe even better than me, about what is a validation of the model we have to, because when we talk about the food supplements, all the compounds which come from that is even more mess than with a drug, because with a drug, we have a little bit more information than concerning all the things what we have. So it's still unknown field in many aspects to do that. And the last but not least, I think that we, as you mentioned already about the sponsorship, I think we should not forget about one of the main drivers here, which is money. So bringing a drug to the market is at least to clinical trials is like 3 to 4 billion euros. And that is very expensive. So basically what we see from pharmaceutical point of view, that if the system could be the helping pharmaceutical company to already put the label, the green label, red label, yellow label on any compound they already used in the lead development process or candidate selection or validation or whatever, that they know that this particular drug or this one should not be, for example, used in pregnancy. Okay, because it's neurotoxic or it's cardiotoxic or it's whatever, but not only because of that, but because it can penetrate the placenta too much. So something like that, which give you an additional risk assessment tool. And I think what we mentioned today several times about in silico part is very important, because one of the instrument what we do, I see here Novella from EngineSoft, and we also have people from INRIM, the Italian National Institute for Metrology, who are developing the process of the multi-criteria decision aiding tool together with uncertainty quantification, which is a kind of mathematical methods used in data processing to understand what is level of confidence, what is level of credibility to do that. So from technology point of view, I see no big problem here. If you have good data, you can have it. What I'm more concerned is, for example, from the doctor perspective. If I would be a doctor and said, okay, and I get this information, said, for example, the drug A is cardiotoxic and it penetrates placenta, presenting the risk of 72%. So what should I do? Should I give it or not? Should I prescribe it or not? So it's also the question we need to interact with the clinicians a little bit more tightly to understand what kind of information they actually need in hand to do that, because their decision at the end of the day is binary, yes or no. So it's very simple at the end. And I think that also in regulatory aspects and all the practical aspects, that is what we need to do a little bit of multidisciplinary discussions. Sorry for long introduction.
No worries, but I think it is really a triggering way that we need to answer this. Going further, how do you see the finding from your projects influencing future health policies and practices?
May I maybe continue here from Michael's intervention directly, responding to this question? You have said that your tool is interesting also in the way that it could provide you just the alert that the drug chemical would be more prone to cross the placenta or less prone. And here I would like to refer to this Designathon approach for classification, where the challenge was really put out. Actually, Designathon was a challenge to the science world to say, so how would you classify today chemicals if you were not allowed to do animal testing? So imagine a world there is no animal testing, you cannot do, you mustn't, whatever. How would you do with all the new technology you have at hand? And what they came out as a first proposal, we'll see how it evolves, is to have something like on the epsilon axis, on the y-axis, you have the indication. So what's the bioavailability? And here's where your model could come into play. So high, medium, low, something like that. And on the x-axis, you could have what's the toxicodynamic effect, so concentrations at low, high, or medium concentration, something like this. So in such a simplified classification system that could be used in the one or the other way, it could transform the globally harmonized classification system. It could be used as a first alert, screening, whatever. Your model could have a good role, I think, and then we could also even relate this to cardiac toxicity. My view. I mean, we whatever. So that would be, let's say, a field where we could use it because, and let me say this also, because for classification of chemicals. So we always use positive alerts, right? So we have a class for specific target organ toxicity or for carcinogenicity. We do not have a class for a chemical is not toxic. So we react on toxicity, on knowledge of toxicity. If there's no knowledge, we don't classify. If there is knowledge that there is no toxicity, we also don't classify. So we don't differentiate. So the classification system is something that provides you alerts, what you can act on. And in vitro models may be even easier to be used in that alert system. You say, so if they are validated for reliability, for relevance, right? Then you could say, so I have here something that triggers an alert, and you could develop new classification systems around that. So here we could have a huge impact on regulation, referring to your question, Puya. Future health policies. So if we can, move ahead this current classification system by developing new, even ideally KHS classes based on NAMs that don't predict even the current animal test, but they show new type of alert that could be used because we are aware with new science, we can give you new alerts. We know about mechanisms, we know about kinetics now, and so on. So that is where I would see a good slot for future impact, changing or not changing, further evolving and deeply changing in that way the classification system. That has impact on transport, and lots of things, storage, not just labeling, storage, worker protection, and so on and so forth.
A quick also comment to you, Martin, is also, I think that is also concerned all chemicals and drugs. For example, chemicals which were 50 years ago think they are okay, but now we find out they are toxic. So and also repurposing of the drugs, because some of the drugs are changing, the dosation changing, the excipient are changing. So it may create a kind of new synergetic effects which are not always desirable. So I think that is also concerning PMS and vigilance process, which every manufacturer actually should follow.
I see a raised hand from Brigitte.
Yes, no, it's just to add as an extra thing to that, I think in that sense, it will also be very important when you're developing your method or when you're describing your method to also get some insights in the negatives, because if you that like almost everything will be positive, then such a classification system will also have no value anymore, because people will say, yeah, whatever will be classified as positive. So in that sense, and that's something just in general also very important, to not forget the negatives when you develop a system or when you want to describe a system, to see if you have a compound that is not cardiotoxic or is not passing the placenta, that your system is also able to show that, because then is also very valuable information to have confidence. So not only showing that your system can show transfer or toxicity, but also the other way around, that it's not just picking up whatever.
And a quick response to Birgit also, that it is exactly the point what we like to address, because thinking that when you know how much some certain compounds you have in the blood flow, then you can link your PK/PD model. Then you can link your toxicology model and say, okay, we have exposure like 1 millimole on that one. So how does it affect something? Or we have exposure on 100 micromole or whatever, and that it may be completely different. You can have NOEL, oil, whatever level, because you don't know just yet how much the things would go there.
Mm-hmm. So going further, my question is for Laura. How do you think that LifeSaver addresses the economic and social aspects of sustainability?
So maybe to start with the social aspects. So of course, as Michael already explained, there's a serious knowledge gap on the effectiveness and the safety of pharmaceuticals and the safety of chemicals in pregnant women. This is, of course, very important because in high-income countries, for instance, 90% of pregnant women take medication during their pregnancy. Actually, over 90% of the drugs lack efficacy or safety information in pregnancy. And Michael already explained to you the reasons why, because pregnant women are excluded from clinical trials most often because animal models fall short in replicating the placenta, but also because there is not really a standardized approach of collecting information on medication exposure during pregnancy and so on. So because of this knowledge gap, of course, pregnant women, if they take medication, this could have a negative effect, but also it can give them stress. Just reflecting on whether or not to take the medication, which is also having negative consequences. But also if they don't take the medication they actually require, it also has negative consequences, of course. So LifeSaver is very much working on this social aspect, and by addressing this very important information gap, of course. And then in terms of environmental sustainability, of course, with this biodigital twin, a combination of an in vitro and an in silico model, we are minimizing the need for animal testing. We also focus on the chemicals, as Michael already explained, and how it affects pregnant women and the fetus, how it helps in making regulatory decisions and so on. And also in LifeSaver, we try to emphasize sustainable laboratory practices. We also look at the waste aspect. We look at good manufacturing practices and so on. So this is in a nutshell a bit how we address the sustainability, the economic and social aspects of it.
I think it's also a quick addition also that we look, for example, for environmental footprint of the in vitro system. So we produce less waste as well. So we don't need to do everything like single use. Something can be recycled, reusable. In microfluidic systems, sometimes it's not possible, but sometimes is okay. And I think also important aspect what we like to bring in the regulative purpose, because most of the assays you use standardized assay like PAMPA for permeability of the barriers. They are very limited because they're using wrong data, wrong conditions, and they, for example, may have only one time point. But if you have only one time point, you cannot have kinetics of the transport. You need to have, so what we like to bring to the attention, so it's not just dose response, but it's time dose response. So the time component is very important to see how the things goes on. When we talk about pharma, it may be like, 3 times per day, so 8 hours. But if we talk about environmental pollutant, which nobody likes to take, but you take that anyway, because if you like to, seafood and then you've got microplastics or whatever, and it's a very low level, but say very chronic. So we need a little bit different protocols to be, and maybe different regulations in terms of what is treating with the environmental pollutant to health and what treating with pharmaceutical products, because they have slightly different, or maybe not so slightly, but different boundary conditions.
Brigitte Martin, how would you like to address it from the project alternative? The economic and the social aspects that connect with sustainability?
Yes, Martin, please start, and then I can always add.
At least I can just comment that already, Martin, and with other sister projects, we already have green practices for Green Deal research projects. So there's a lot of work already done. But yes, Martin, please take the floor.
Indeed, you could link out to our policy brief on how these Green Deal Health Cluster projects, where also LifeSaver is part of, have a positive impact on sustainability, and sustainability integrates really economic with environmental and social impact, right? So you could read this. I would say Laura has really talked about everything here in my view. So maybe I would like to stress that really reducing the ethical conflicts that we have with animal testing could substantially improve the social sphere of sustainability. So a positive effect on society. We reduce this problem, let's say. And then I would hope that we find some market opportunities also for this new NAM-based approach. So market opportunities on the one hand for the chemical industry. So being able to really produce and then also assess green chemistry, which is a new chemistry, which is maybe built on renewable resources, not on oil resources. So this will be easier for them to develop this new green chemistry, and hopefully also the testing and assessment services will have growth possibilities. So using computational and complex NAMs is not something it's something you can offer. So just 2 points. Thanks. Over to Birgit.
Thank you.
Brigitte,
go on. I can see your hand.
I also just wanted to add that specifically then in the case for cardiotoxicity testing, as I've also mentioned during the presentation, this is currently not really addressed in a consistent way. And as a result, it seems that it's not really a major issue for the chemicals, but on the other hand, it might be that it looks not being a major issue because it's not really investigated. So in that sense, I think by bringing these new tools, which makes it also more easily accessible, will give us also much more insights in how big the problem is of cardiotoxicity induced by chemicals. I think this is something that we currently don't have very well on the radar, because it's like a bit the catch-22 situation where you don't know, is it because we don't investigate it, or do we not investigate it because we think it's not there? So I think in that sense, it also will have an important societal impact, because as also shown in the presentation, cardiovascular diseases is really an important cause of death among the worldwide population. So in that sense, it will also have an important societal and as well, of course, as an economic impact, because if we can reduce exposure to chemicals that might be leading or contributing to cardiovascular diseases, this will also have a huge economic impact in the end if we can reduce the occurrence of this type of diseases.
I think it's also in Japan that issue is addressed as trilemma in the society. What we can say in European, if we translate it to Europe, is we have here the chemical producers. So they are not interested to know if their products are toxic, because they want to sell that. And if they sell something toxic, they are not happy about that. So they have their own economic drivers. On the other hand, we have society at large, because people does not want to eat toxic chemicals. Of course, who would like to pay that? So we have a combination of that one. And usually it somewhere stacks, somebody who is lobbying that or that, saying, is it toxic or is it not toxic? And then it will be toxic known after 20 years, but I'm retired already, I'm not care, and so on. So I think that the role of regulators here needs to be stressed, because if we can give to the regulators a correct method, they can justify and said, okay, with this method, we know that this is something we can confirm whether it's toxic or not. And if they import it by regulation, so if the company producers want to go to European market, let's talk about European market, or even in US market, they have to fulfill certain criteria, because otherwise they cannot sell the product here. They cannot manufacture that, they cannot think. The REACH was trying to do that, but REACH is very sluggish and it takes a lot of time, and still 75% of all chemicals are not even classified, as Martin mentioned. So, or they don't know how to classify it, or they don't know how to validate it, and so on and so on. So I think the role of regulators is a third player in these two, between society and industry. They need to understand what they're doing. And for that, our role, at least in LifeSaver, how we see that, we need to provide them policy advice. We need to tell them what is needed to be done, and that needs to be told in a scientific, grounded way. Not just, okay, we develop a system which says test you like that, so here it is. That is not what they want, because it's need to be explained to the member of European Parliament. It's need to explain to the STOA committee in EP, in the council, and and then it starts moving, because otherwise it's maybe not working well. Martin?
Yeah, and immediate reaction to this, I fully agree with you. And I think one useful way to move ahead could also be to focus or to develop classification approach, which is really based on potency. So where it's not, this is cardiotoxic and this is not, or developmental toxic or not, but where we say, well, this does it at really low concentrations and this only at very high. So let's care for the one with a really low concentration. This could be a place where we meet with industry, regulators with industry, because this is a bit of a hurdle. Yeah, you get the stamp of being toxic and you're out, and in scientific reality nothing exists like toxic or not toxic. It's first all of a probability and a second on dose. So we need to get the science in here to inform regulators that it's a dose issue.
Right. And beside that, we also need to know how do we reliably measure that? Because if you said some low dose, what is low, and is it limit of detection of the recent technology, or if you need to use a complex, sophisticated method of analysis where one test would cost like 5,000 euro, nobody will do that. That is for sure. And this is, I think, a classical example. What you take is 4-hydroxynonenal, because it's a very, very non-toxic product of the lipoperoxidation of the fatty acids. But if you have only 2 micromoles, that is essential for the cell proliferation, which is very well known. If it's 20 micromoles, that's became toxic, and 100 is completely kaput. So basically, then when we talk about the dose, it's also the question, how did we do the exposure? What is a chronic exposure? Is a chronic exposure on 2 micromoles is equivalent to acute exposure to 10 micromoles? That's also the thing, and that's what I mean. Our project, maybe we cannot be able to solve it till the end of the next year, but we need to develop some consistent methodology to how do we explain it, what is need to be taken into account, what is a priority in terms and explanations that the regulator will understand, because the regulator needs to tell it to the authority which actually stamp the new regulations. And if the Euro Parliament will not understand that, they will not accept these regulations.
Thank you to all the participants, and for such deep and dimensions that I could see with our discussions. We could cover the regulatory part and what needs to be done in future, what could be the potential recommendations, and so on. So thank you once for all for joining, and let's hope that the work that is being done in the project Alternative, LifeSaver, and other sister projects promote safer chemical use and regulation. And we see how we are advancing further with non-animal testing methods in overall improving public health. And let's see if we find some more synergies on collaboration on EU policies that we just discussed could be recommended to the regulators. I think we have come to the end of the discussions.
If I have just a little bit of comment as commenting to what Martin was said, what do you think for the next steps? There could be that if, for example, in the next spring, we can together write down some kind of policy draft, because I think, Laura, what we're doing in LifeSaver, doing some policy advice, which has to be like, 2-3 pages paper, where we highlight how exactly and what needs to be done, what Martin was and Birgit was mentioned as well. So if we need now to put it on paper, it can be just a bullet points or something as a draft, but I think that would be great that if we can cooperate together to create some kind of approach of methodology. You can call it position paper, you can call it in a different way, but I think that would be one of the great also deliverables what we can do together.
Definitely, it is an idea that could be explored, but just to let you know that the project alternative is wrapping up in March. So if we can start early in January, maybe then it would be more helpful for us also.
Deadline. Yes, that is a deadline.
Yeah. So good, then I will...
Maybe just as a suggestion, Pooja, you can ask because you in WebEx, you have the AI assistant, which can make like a report of what was discussed here. You have the recording, so what was discussed during this meeting. So maybe this can then be used as a kind of starting point for making a kind of report on it.
Yeah. And that's why if you can iterate it like, before Christmas to send us so we can comment among the participants of the workshop. It can be like open document, and then we can squeeze it within 2 pages, saying that this is a joint position of the Green Deal Sister Project, which we can deliver to the Commission and say that is how we see that things should be done.
Sure, great idea, and I'm really happy we are coming out some call to action items here and look for future collaborations. Thank you all for collaborating. May we all switch on our cameras for a group photo, and this will help also in dissemination and will give some impact created by this workshop. Can we have other participants who are in the call could open their cameras and we could click a picture?
Maybe not everyone has a camera.
Okay.
Okay.
Okay. No worries. So anyway, I have the glimpse of all the panelists and the names of the participants. If no one has the objection, then I will share it on LinkedIn and I will tag few of you. Thank you, and it was really great having you all. Until next time, take care, and we stay aligned via our communication. Bye-bye.
Okay.
Bye-bye.
Thank you.
Thank you.
Thank you.
Bye.